Thank you so much for bearing with us in that community building lunch event. I hope everyone made it there and back all right. And now we have a little treat for you before we move into the next session. Barry Levy and Greg Lagana, who have done their damaged care presentation for us 2 years ago, have graciously decided to kick this off because this part of their show is so fitting for our next session. So no further ado.
Good afternoon. So instead of being human beings here at the 8th Annual Gold Lab Symposium, imagine for a moment that you're at the 8th Annual Conference of Microorganisms. You're all microorganisms. And you're at the 8th Annual Conference getting together, plotting against everything you're going to hear this afternoon in the formal presentations here at the Gold Symposium. So bear with us. It's great being here at the Conference of Microorganisms. Everybody's looking so great. The bacteria.
Hi.
The viruses.
Hey.
Even the fungi and the molds.
Jeez. Everybody came, but you know, it's getting really difficult to recognize each other without a microscope these days.
Sure is. Hey, how's your family doing?
Oh, I am proud to say that the Staff family is alive and well.
The Staph bacteria family is alive and well. Didn't vancomycin almost wipe you all out?
Well, almost, but not quite.
I heard that 99.99% of your family just dropped dead.
Well, that's true. But that still left me with 10 surviving children, 10,000 surviving grandchildren, and 10 million surviving great-grandchildren. Want to see the pictures?
Just put them up on Facebook.
No. I now have well over 1 million trillion great-great-great-grandchildren. Want to see their pictures?
All of them resistant to vancomycin?
Yes, and resistant to just about every other antibiotic known to man.
What a time to celebrate.
Me bugs have much to be grateful for.
Multiply resistant TB down in Denver.
Rocky Mountain spotted fever right here in the Rocky Mountains.
Yeah, Lyme disease in Connecticut.
Mad cow disease all over the place.
And look, in the Wall Street Journal today.
Ooh, e-business.
No, E. coli.
Another outbreak of a super bugs.
Bum, 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 bum. Another outbreak of super bugs. An epidemic, come on, some hugs. Another failure of wonder drugs. Another outbreak of a super bug.
Salute TB,
A resistant group. Salute the strep, they turn flesh to soup. Salute E. coli, they live in poop. Another outbreak, what a snazzy group. Go ahead, give us nice Latin names, but we will get you quite just the same. Invading your body's our #1 goal. You can't cover up each and every hole. We're always launching some brand new schemes. We're scaring parents about kids' vaccines. The measles virus is on the rise, 'cause parents don't get their kids immunized. In this age, we travel in style, and we earn many a frequent flyer mile. Yesterday, far away, one of us rose a jet flight today, and we're in your nose. Against us bugs, no security. So don your mask and call CDC. Wear gloves, wear condoms, use DDT. You might gram stain us, but you can't contain us or our family. Bum, 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 bum. Hey!
Thank you. We love you, Boulder!
We can save the questions for later. That was excellent. And I'm supposed to say, on our website, you can view the entire Damaged Care show, which was performed at the Gold Lab Symposium 2 years ago. So we have a legacy of wonderful performances. Thank you so much. I want to personally thank you because as moderator, I was racking my brains to think of something funny to say about today's topic of this particular part of our symposium, because a lot of it is not very funny, and so, you know, now I don't have to do that. Thank you. So then instead I thought, well, then I should say something kind of random and personal about the symposium after the dog bites thing from Charlie. So that's what I'll try to do. So I'm Anna Pyle. I talked here 2 years ago, and I study RNA structure. I come from the RNA world here at University of Colorado. Right now, part of my lab studies how RNA molecules are recognized by the surveillance system of your innate immune system and the proteins in your innate immune system that recognize things like flu and flaviviruses and other nasty things. So my stuff's really very molecular. But today we're going to talk about a broad range of interdisciplinary topics in microbial disease and its sort of threat to us globally. And so what I wanted to sort of say that I had experienced personally that sort of condenses the topics of this symposium come from my days as an undergrad. So I was a teenager in Albuquerque, where I grew up, and I spent my childhood mostly skiing and going to parties and being silly. And when I showed up as a freshman at Princeton, I just really was basically unable to function. I had no study skills. I didn't know how to do anything, and I nearly flunked out. But there were a few professors there who took an interest in trying to help me learn what I needed to know to get through school. One of the people who was really key there taught me to write a research paper, and he was a historian named Arno Mayer. And he's one of the foremost scholars of modern Europe. He was actually a spy in Germany. He was kind of a secret agent there during the war, very brave guy. And so he intended on ensuring that I would go to graduate school in history. And I loved history. And then one day I had to go to his office and tell him, Arno, I'm not going to major in history. I'm going to become a chemistry major. And he just completely flipped out. He was so angry. And then he said, I consider this a personal betrayal. So I was like, I'm so sorry, Arno. Anyway, Arno and I remained friends throughout, for many years. And about 8 years ago, I called him up because we would stay in touch. And I'm like, Arno, how are you? He's like, I'm fine. How are you doing, Anna? I hope you're giving them hell at Yale. I was like, well, I'm trying. And I was like, what are you doing? And he said, well, you know how I was very disappointed that you didn't go into history. In fact, in this later stage of my career, I'm reading a lot of articles about bubonic plague, and which is Yersinia pestis. It's a bacterial infection. And he said, the reason is that I've spent my whole career studying modern Europe, and now I think that the foundation for everything that we experienced, all the trauma of the 20th century, came from the Middle Ages and the Black Death that passed through. So I'm wondering how many of you have read the Doomsday Book by Connie Willis? Anybody raise your hand who's read the Doomsday Book? There's only one back there. Oh, there's some back here. This, we like to share reading that we've enjoyed, and so that's my suggestion. Read the Doomsday Book. It was written in 1992, and it's a classic piece of fiction. And what it is when a historian, there's a time machine, and a historian from England is sent back in time to be a witness to the development of the Black Death in England. And so you see through this very personal lens the crisis that emerged within families, within communities, and then eventually engulfing whole cultures when that disease ravaged Europe. And it seems very abstract to us at this time, but you have to realize there were parts of France that were completely depopulated, where nobody survived at all. And these were wealthy regions of France, and so the culture did a complete flip. Groups of people that previously had no access to power suddenly filled the vacuum. All of the relationships amongst groups of people changed. Europe utterly changed. The reason I bring this up is because this particular part of our symposium is going to be about microorganisms, the threat of global health, but it's also going to talk about the connection between global health and its influence on our culture, on the interactions between species, and then reflecting on this is how we perceive our responses to that threat in a more holistic way. So in the end, that's sort of my hope, is that you'll see all of this broadly and you can have a more personal connection to it. So our first speaker today Now I have to pull out my glasses. Well, first of all, let me say, we are just incredibly, incredibly lucky to have Julie Gerberding here with us. She's one of the people in this field I admire the most. She's probably most famous for being the former director of the CDC, and I believe you started there in 1998. When Julie started there, she initiated a tenure over about 10 years where our country experienced some of the most formidable threats to public health that we'd ever experienced, and some of these we hadn't even yet imagined. And Julie was at the helm during that very scary time. Prior to that, though, she was a professor at UCSF, where she was director of the Prevention Epicenter, which developed strategies for preventing infection in patients and providers. And if you think about it, during that time, San Francisco was a very interesting place to be doing that job. Right now, Julie is at Merck, which she joined in 2010 as the president of Merck Vaccines. And now she's an executive VP of something that has a title that's just so long, I don't think I can recapitulate it. But basically, global public policy, population health, and a variety of other things that have to do with communications and global health planning worldwide. So without further ado, thank you so much, Julie.
Well, thank you. It's very exciting to be here, and I appreciate that warm introduction, but I've never had a warm-up act like that before. So if y'all need a booking agent, I think there's some boring infectious disease meetings where it could really liven up the conversation. Okay, it's a deal. I'm going to try to keep in the spirit of today's conversation and talk a little bit about antimicrobial resistance from the standpoint of the evolution of our understanding of what's really going on, asking those why questions. But why does this happen? But why does this happen? Digging a little bit deeper. But I think to start out with, just to make it clear, I think I need some help getting my pointer on, making it clear that just today, the CDC issued a new alert about a new drug-resistant Candida species, Candida auris, which has been around for a while. It's a very serious infection in healthcare environments. It causes bloodstream infection, wound infection. It's called auris because it was originally isolated in the ear, but it's a Candida species. It's basically resistant to all three classes of antifungal medications. So even as we're speaking, there are new problems emerging, and the urgent threat level is being raised once again for these organisms that we're just simply running out of drugs for. So this really is a global health problem, not just a problem for sick people in intensive care units, but a problem for everyone. So if you want to keep up to date with the data on what's emerging and what it's sensitive and resistant to, of course, the CDC remains an excellent resource. But I am pleased to say that these activities have now globalized to the point where governments, the G7, the G20, is meeting as we speak through the health ministry, heading into the World Health Assembly, and antimicrobial resistance is finally at the top of the conversation. So we hopefully will have some concerted global action on this front imminently. So what I'd like to, again, start with is a little bit of the evolution of our understanding of this problem, really starting with version 1.0. So 1.0 really started, of course, with Alexander Fleming, who discovered penicillin. And I thought this was a good introduction because he was really fishing, right? He didn't know what he was looking for, but he fortuitously discovered that the Staph aureus on his petri dish stopped growing in the vicinity of the penicillin mold because they were being lysed by the soluble penicillin that was being released there. And instead of tossing the plate out and moving on, he asked questions about why is this happening, what's going on here, and went on to really understand what penicillin is and did. But almost simultaneously with his discovery of the compound, he also discovered penicillin resistance. This is taken from his Nobel Prize lecture in 1945, but even in the early lab experiments, they were able to identify organisms that were resistant to penicillin. And he created a hypothetical example in his Nobel address. Supposing that a patient is taking penicillin, doesn't take it in the right dose, has a subtherapeutic concentration, then passes that resistant organism onto his wife, who gets pneumonia, and then, of course, she dies because her pathogens have become drug resistant. So almost well before penicillin was being used clinically, he understood the potential for this problem. And this really was, for a long time, our understanding of antibiotic resistance, that in a population of organisms, there would fortuitously be some lucky bacteria that were resistant to the drug, and under antibiotic selection pressure, those lucky organisms would have a chance to survive when everything else was killed off. And assuming they hadn't lost any fitness as a consequence of their mutation, they would then exist to repopulate and become the dominant organism, just waiting for the next time an opportunity to cause disease comes along. I've had many opportunities to read about this kind of drug resistance, but I had one opportunity to watch it unfold in action. And that was during my time in San Francisco when, because the city was so hard hit by AIDS, and San Francisco General Hospital, where I was working, really was the epicenter of AIDS in the city, we watched what happened when a very large proportion of AIDS patients in San Francisco were started on Septra prophylaxis for pneumocystis, toxo, and other opportunistic infections. So on a population level, an enormous number of people were suddenly put on daily broad-spectrum sulfa drugs to try to protect them from these opportunistic infections. This is probably the best research I've ever done. I think it would be a candidate for the Parasite Research Award because we were using secondary data sets. But it's also really important to me because the second author on this paper is my husband, and I probably wouldn't have met him and gotten to know him so well if we hadn't worked on this project together. So it's a really fond memory. But what is shown here really in the green bars is what happens to isolates of bacteria cultured from these treated patients over time, over an 8-year period of time in San Francisco. And you can see we watched the emergence of Septra resistance right under our eyes. The non-HIV patients also had some increase in Septra resistance. We think that's because we didn't know all the AIDS patients. If they weren't housed on the AIDS ward or being seen in the AIDS clinic, we probably misclassified them. But we were watching an incredible upsurge in resistance. Not surprising, and this, of course, was all based on selection for the relatively frequent point mutations and then a replacement with the resistant organisms. And then what was obviously going on was gene transfer, but at the time we weren't recognizing that, which leads me to the second evolution in our understanding of antimicrobial-resistant version 2.0. So this Nobel Prize winner is Joshua Lederberg, who is just an amazing man. I knew him well and had many opportunities to interact with him. He was an amazing champion of antimicrobial stewardship and an outstanding scientist. But he won his Nobel Prize for his very early work, which demonstrated unequivocally for the first time that bacteria can exchange genes horizontally. Up until that point in time, this was regarded as impossible. It was almost scandalous to suggest that it could happen. But in a simple and very elegant experiment, he took 2 strains of E. coli. One was deficient in its ability to manufacture 3 amino acids, and the other was deficient in the ability to manufacture 2 other amino acids. But when he mixed them up, they were conjugated, and ultimately in the middle column you can see that some really healthy colonies of E. coli came out of that because they had picked up the normal genes from each other and were able to thrive even in a media that was not permissive. So he went on to really establish horizontal gene transfer mechanisms in a number of directions, which I've shown on this cartoon as a very simple model where a sensitive organism mates with a resistant bacteria, picks up the genes that confer antibiotic resistance either by free DNA in the media or by conjugation, which involves cell-to-cell contact, or transduction, which involves a phage to translate the DNA from one organism to another. But regardless, what you end up with are organisms that over time can accumulate a number of drug-resistant mutations through this horizontal gene transformation. And I love the statement by Julian Davies, who's probably my favorite microbiology expert, who basically says this happens all the time everywhere you put bacteria together, and it's a virtual bordello. They are so promiscuous that they are constantly exchanging their genetic material, and so there's a universal opportunity for these genes to move around. And ultimately then, again, in this cartoon format, under antibiotic selection pressure, you kill off the susceptible organisms, even if you're using a very broad spectrum drug, and you end up with resistant populations that have multi-drug resistance. Now, that's part of the story, but why is it that these multi-drug resistant organisms are so concentrated in the healthcare environment? Well, obviously the patients are sick, they are prone to infections, they have a lot of antibiotic exposure, and so on and so forth. But there are aspects of healthcare environment that are particularly permissive to the emergence of antimicrobial resistance, and that has to do with the presence of biofilm. For those of you who aren't familiar with biofilm, this is a scanning electron micrograph of a biofilm in a urinary catheter. But anytime you put something artificial in a body fluid, proteins, carbohydrates, complex polysaccharides, things stick to it, including little bacteria that normally would transiently maybe pass through and be eliminated by your immune system. They are able to hide out in this biofilm and escape immune detection and the other normal host defenses. The biofilm itself promotes the emergence of antibiotic resistance by virtue of its sheltering of the bacteria that are hiding in there. And they begin to build up very unique and specialized colonies of organisms, many times different species of organisms in the same biofilm, and they communicate with each other. They're usually not growing very fast because they're not seeing a lot of nutrition, so they are slow growing, and when antibiotics are presented to the biofilm, often the drug can't penetrate to get down to where the bacteria are, so they see sub-inhibitory concentrations of the drug, which might slow them down a little bit but doesn't kill them and allows them to adapt to that and ultimately escape. So biofilm promotes antimicrobial resistance, and not surprisingly, the pathogens that are most often seen in association with healthcare drug resistance are those that are also associated with biofilm and device insertion in patients. The prototypic organism here is, of course, Staph aureus. We've already heard about that in our musical, but the Staph family acquired widespread penicillin resistance fairly quickly after penicillin was introduced during World War II. By the mid-50s, roughly half of Staph were resistant. By the mid-60s, virtually 90% of Staph were resistant. Methicillin came along in the 70s and kind of saved the day, but very quickly MRSA, or methicillin-resistant Staph aureus, emerged, which is now assumed to be present when you have a staph infection in the hospital. In the 90s, we began to see vancomycin resistance in a related organism, Enterococci, and then some moderately resistant Staph aureus in organisms called Visa. And ultimately, and fortunately only occasionally so far, we see full-fledged vancomycin resistance. So we have penicillin, methicillin, and vancomycin-resistant Staph aureus, and hence the superbugs that we were just charmed with. And we do have new antibiotics that are effective against Staph aureus, but we have already seen emergence of drug-resistant isolates in these categories as well. So this is where the pearl that medical students are taught emerges. And the concept is, if you lose it, if you use it, you lose it. Meaning, anytime you use an antibiotic, eventually you will lose it, because the bacteria, sooner or later, will figure out how to express resistance to it. So obviously, this is a situation where less is best, and leads to the whole theories and principles of how we should have better utilization of the antibiotics that we have and never waste them or use them unnecessarily. Now, one of the things about these superbugs, they don't just stay in the hospital, they like to travel. And they travel in a lot of ways. They travel, of course, in a community by virtue of the fact that patients don't stay in the hospital. They go to the nursing home, they go to their home, they have care in a variety of different settings. But these organisms travel globally. They do that through activities like medical tourism, where a patient in India may come to the US for a procedure and introduce a resistant organism here, and vice versa. Our patients who go to other countries for medical care bring their bacteria with them, introduce them into healthcare environments. We also see examples where travelers who are healthy can acquire drug-resistant strains. There's an elegant small study from Sweden where some Swedish students who were dispatched either to India or to South Central Africa were cultured before and after they went, and they were very clearly able to demonstrate acquisition of new resistant genes among the flora that these students were colonized with. And some of them were gone for no more than a month. So these were a few weeks of exposure into a new environment, not a few years. So what has ended up happening is that we have a resistome of resistant genes that are truly globally distributed. This is a map of just one kind of antimicrobial resistance gene, one that's pretty deadly because it makes it impossible to treat very important nosocomial gram-negative organisms. But you can see that the patterns of these are not the same in every country, but by mapping them sequentially over time, you can see how they emerge in one part of the world and then eventually travel to another part of the world and then replace whatever was there before, and so on and so on. So this is a global issue in developed and in developing countries. So now let me evolve a little bit further and ask the question again, why do antibiotic-resistant pathogens emerge? Well, in this case, we're going to talk about a different Nobel Prize winner. This is Selman Waxman, who is a soil microbiologist, a Ukrainian scientist who came to Rutgers and was very interested in the soil, its composition, its relationship to food production, and ultimately the bacteria that were in it. In keeping, again, with the spirit of some of our conversations this morning, he was not a scientist that was overly prone to credit his collaborators. So there is some controversy about his claim to have discovered streptomycin, but he and Albert Schatz, as well as a number of other collaborators, really systemically studied soil, some of it right from Rahway, New Jersey, and were able to find all kinds of soluble chemicals in the soil, many of which had antimicrobial products, and a few of which were non-toxic enough to actually be developed into human drugs. So particularly streptomycin, which was so important for tuberculosis in those days, as well as neomycin and actinomycin, which is an anti-neoplastic agent. So he really introduced the notion that there were sources of bacterial compounds in nature besides penicillin mold that could be extracted and manufactured for purposes of preventing or treating infections in humans. When you really dig deep into the science of the soil, you discover that it has got to be one of the most fascinating and least understood niches in biology. This is just a picture of grass in a cross-section, but you can get a sense of how complex the structure of dirt is. It has minerals, it has air pockets. If you look at this cartoon of a little clump of soil, there's the air gradient, the oxygen and nitrogen gradient. There's the nutrient gradient. In here, there are gaps where little niches of bacteria, micropopulations, can situate themselves. There's a gradient along the rhizomes of the roots where the nutrition is high and favorable to bacteria, and other places that are dead zones where the bacteria can't really live. And what we understand about this environment is that it's incredibly complicated, even in just a handful of dirt. That the microbes are spatially concentrated in certain areas and not in others, and that there is really a chemical cornucopia of organic molecules that are being produced in this environment. We can't measure them, very easily, because often the concentrations are so very low and it's just very difficult to extract enough material to understand what's really going on. So we were kind of stuck there for many, many years. But of course, now that we have metagenomic capabilities, we can go in and study the molecular construction of the bacteria that are there. Many of the important soil bacteria have been sequenced. We're just beginning to really see from the genetic perspective what really could be going on there, and beginning to create models and environmental opportunities to have a little bit better look at what these bacteria are already doing. But one of the fascinating things is that we know that these soil bacteria have a very high frequency of resistomes, or genes that do confer resistance to antibiotics. And they do that through either functional or metagenomic sequencing, where they, for example, chop up the DNA in the bacteria, clone it into E. coli, and then see if the E. coli survives antibiotic exposure or not. So they're developing a much deeper understanding of what is really going on in the genomes of these organisms. And that leads to the concept of the parvome. The parvome being the bioactive organic molecules that occupy a particular ecologic niche. In these environments, you can have bacteria that are secreting small molecules, and these molecules have lots of functions, probably most of which we don't even understand yet. They can signal other bacteria to either grow or divide or excrete something. They can send out quorum sensing signals so that the neighborhood understands who's there and what population and who they have room for and who they don't. They can serve as nutritional sources, carbon and nitrogen sources for other microbiome neighbors. But they can also mix up with the same sort of particles or same family of particles that are coming in from other bacteria, so that in the end you end up with an extremely complicated chemical milieu. And of course, some of these organic molecules are toxins, and they are a defense mechanism against competitive neighbors. Now, if you think, bacteria have been around for billions of years, you have to have some defense mechanism to keep your neighbors from taking over your niche. And so what we might refer to as an antibiotic, because it's important for humans, actually in the context of a microbial community, is just a defense mechanism, leading to the notion of the arms race that's going on between bacteria. They need to be able to make molecules that are toxic to the competitors, but they also have to have the genes that code for molecules that resist that. So there's constantly a struggle going on. And if you're making a potent toxin that can blow somebody up, you probably want to make sure you have something in your genetic storehouse that will keep you from being blown up. So this dance that's been going on for millennia has really enriched these populations of antibiotic resistance genes from our point of view, but really defense mechanisms in the soil. Now, we know a few things. We know these resistance genes evolved long before antibiotics were created and used for human use. And we know that the environmental resistome that comprises this entire genetic mass is large and actually probably covers all known human antibiotics. And let me just give you an example. This is an article where some investigators found a rare bacterium in a cave that had not been exposed to anything or anybody for 4 million years. And they were able to sequence this bacteria and found that it had genetic markers of 18 antibiotic resistance genes. So it was there long before that bacteria needed to worry about vancomycin. They also, in a separate study, were able to show that molecule that I showed you had globalized, the ESBL, or extended spectrum beta-lactamase, CTM gene type, was probably contributed by a soil bacteria called, I think it's pronounced Clivera, who had never been exposed to antibiotics, but the gene in the bacteria in the soil and the gene that is circulating globally in healthcare environments is virtually identical, suggesting that that bacteria donated that gene somehow. It made its way into the hospital bacteria where it has conferred a selection advantage and under antibiotic pressure has become dominant. We also know that the more we look, the more we find examples of genetic identity between the resistant markers in human pathogens and soil bacteria. And as I said, we have at least one example where there's been clear-cut evidence of pretty recent transfer. So this resistome in the soil and the resistome in the hospital among pathogens is pretty similar. Now, there are a lot more of it in the soil than we are yet seeing in the hospital, but there is significant overlap. And the question is, how did this happen? I mean, some of us play in the dirt, but this should really not be a common occurrence among hospitalized patients. So obviously, we're missing a piece of the story here. And I think the real breakthrough in understanding is where are antibiotics really being used? And antibiotics are really being used in food production. Nobody actually knows how much antibiotic is being used for growth promotion, treatment, or prevention of disease in farm animals, livestock, poultry, or salmon in aquaculture, et cetera. But in the United States, the FDA estimates that at least 80% of all antibiotics produced are used in the food supply. Not in the hospital or in people, but in animals and fish. So, of course, what happens with livestock? They poop. That goes in the soil. So all of the opportunities that they have to have selected for resistant genes in these animals under long-term exposure to antimicrobials ends up in the soil. And then horizontal gene transfer works in the opposite direction, and the animals are able to benefit from these resistomes that they're picking up in the opposite direction. Likewise, people eat food. So we are constantly exposing our intestinal tracts to the resistomes of the food sources that we eat. So we've created a very nice ecological cycle here where we are putting an enormous and unprecedented selection pressure into the system. What's going on in the soil with these tiny little sub-detectable levels of antibiotics on an ongoing basis is nothing compared to the tons of antibiotics. And the fear and the worry that's exciting ministers of health and others in the world right now is that as the world grows and people come out of abject poverty, there is going to be an increasing demand for protein food sources. So the pressure to improve the productivity of the food supply is intense. And you have countries like China where right now, if there's not a change, the amount of antibiotics that they will need in their livestock production facilities is expected to at least double by 2030. So this is a really important global pressure on the emergence and spread of antibiotic resistance. And this is a more detailed picture just showing that I made a very simple cartoon, but this has impact on our water supply, has impact on seafood. Really, the entire ecosystem is affected by the impact of this kind of antibiotic exposure. So that, in a nutshell, is pretty much how we have arrived in our understanding of antimicrobial resistance so far. Clearly, we have a lot to learn about the parvome and the resistome and these interactions. And I think we're on the brink of really making some progress here because of the technology breakthroughs and the bioinformatic capabilities and our ability to model and understand these systems in much more detail than we could have even 5 years ago. But what we really need urgently are new molecules. And we really have to think through how we change our approach to either using antibiotics or how we change our approach to influencing the small subset of pathogens in a more selective and targeted way so that we're either not destroying their ecological environment by using lasers to maybe turn off their virulence factors or affect their quorum sensing, et cetera, rather than blowing up the whole community and then waiting to see what thugs will come in and replace the bacteria that we have eliminated. So we have to exploit this genomic understanding that we're beginning to fathom in ways that target new approaches, not maybe bactericidal approaches, but new approaches that are much more specific and precise in how they influence the bacteria. And I will just give you one example of a solution that we've needed for a long time, because most of the things that we're talking about of interest, you can't culture in the laboratory. That's why we don't know very much about them. But this is an example where, just in the simplest way, somebody figured out how to take some bacteria from the soil that we couldn't culture, but embed them in single-cell plates, chips basically, and then take those chips and stick them back in the dirt, so that really the bacteria were back where they belonged in the environment that they were used to. And they grew happily in there. And then they could be taken out of the dirt again and sequenced and analyzed. And using this approach, they have found a new class of antibiotics with a new target. We don't really completely understand how it works. It's active against gram-positive organisms, seems to be safe in animal models. So I'm hoping this is just the beginning of the future of what we might be able to do and understand as we bring these different points of view together to bear on the problem. But just in case that doesn't work, I found this on the internet. In case of an apocalyptic event, you can make your own penicillin. And they really give you the recipe. So just so you know, if you don't mind taking some risk with mold in your kitchen, you can extract your own penicillin. And the article was full of all kinds of caveats why you really shouldn't do this. So I'll stop here and take any questions or comments you might have.
That was wonderful, Julie. One thing you hear a lot is, why aren't the pharmaceutical companies doing more? And so, I know you could talk for an hour about that topic, but what are a couple of things we should remember when we hear that?
Yeah, I think there are 2 points of view on this. My view is that we are actually, at least in my company, we have a pipeline and we are investing, and we've got some deep ideas in our pipeline, and we've got some things in phase 2, 3 clinical trials. But the problem is that we have to go from a model where antibiotics were blockbusters and were guaranteed to pay for their costs as well as make money, to a point now where they really need to be used like orphan drugs. So we shouldn't be marketing them, we shouldn't be selling them and making lots of money, because these are drugs that we don't want to use. And so we have to change the reimbursement model so that they operate like valuable treasures instead of things that you can go to the drugstore and get anytime you want to. But I think the real problem is targets. We really don't have any new targets for these drugs, except maybe the one I just mentioned. We haven't even figured out what the target is there. So the science is not leading us to find the opportunities. Almost everything that has been developed since the 80s has been done through modification of existing entities and more pharmacologic innovation than through drug discovery. And that's where we need to bring the power of our genomic sciences together and really hone in on new opportunities there.
Question back? Oh, sure.
A follow-up question. So I've been listening to pharma guys say what you just said for a long time. We don't have targets. And I think I, as an amateur bioinformatics guy, could find in a week 195 proteins that are essential in bacteria that have no human homologs and would be perfect targets. And so
Like a job.
no, no is the answer. But so I obviously have a misconception. Would you say one more sentence or two?
Part of the problem is that we cannot culture these organisms. So everything has to be done by sort of modeling and hope. And until we have either animal models or some kind of system that we can actually test that is relevant to mammalian cell biology, we're a little bit stuck. And that's why this iChip is so exciting, because at least we can grow organisms that we haven't been able to grow before. And hopefully after we have better understanding of the metabolic pathways and how these small molecules are being created, we might be able to understand better how we can culture them. So we have more sophisticated approaches to solving for them whatever it is that's missing from the conventional approaches so far.
I'm going to take a question over from here. She hasn't asked one yet.
I just wanted to know, do you see a resurgence in phage therapy, either here or in other parts of the world?
Yeah, phage therapy is definitely, especially in Russia, there's a lot of interest in using phage, which are highly specific for certain bacteria, and often a phage, which is a virus. I always think of them like little lunar landers, but they will specifically land on a bacteria in a lock and key mechanism and then blow it up. So, that's definite, especially for intestinal pathogens. I think that's important. The other piece that fascinates me, and if I had to start all over again, is what I would be working on, is the biome. There is so much richness in our intestinal tract and in all of the body surfaces that are colonized, the ear biome, the lung biome. There's so much we don't know about our true microbiology. And we really almost biomes that happen to have people wrapped around them rather than people who happen to have biomes. So there's just so much fantastic science that I think we will be finding there.
This one over here, he hasn't asked one yet.
Following up on the biome question, do you think it's possible that this sort of basic germ theory, version 1, has sort of really distorted the way we think about this problem to the point where we're not approaching it correctly? And given that 99.99% of the bacteria in our body are symbiotic and healthy, and given that the entire metaphor, the way we've approached that, is nuclear war, is there a way we could think about how do we create an environment that has a lower probability of transition to pathogenicity, one that produces, at some level it's a simple metaphor, a healthy field is unlikely to grow weeds, and we make ourselves very unhealthy. So obviously we need a new generation of drugs, but then we're going to have the same problem again and again, and I think that maybe we're really under-resourcing the science of what would create something where these pathogens are just less likely to convert. And because I'm just very conscious that this whole field has sort of produced this profound global damage for rare but significant gain. And the balance feels wrong, and even continuing down that path feels wrong.
We're forcing the evolution to work against us rather than for us, and that's what I'm trying to get across in the sense of let's respect the biome and not do things that cause such extensive perturbation that we end up with survivors being only the bad actors. So we have to think of it different. My husband and I are growing a meadow in our yard, and what you said about the weeds is exactly right. A healthy meadow, the lovely perennial things will outcompete the weeds. And you don't add fertilizer, you don't add weed killer in there, you just let the healthy population do what it's supposed to do. And I was really thinking about that when I was looking at the meadow, which is hopefully going to be good this year. It has kind of looked like a weed patch for a while, but and I think, again, the science of what actually is going on metabolically in the healthy bacteria should also be very helpful to us. So there have to be better solutions than nuclear bombs.
She's wanted to ask a question. Yeah, go ahead.
Thanks.
So I became interested in antibiotic resistance about 3 years ago. When looking at Parkinson's disease, I noticed a correlation in antibiotic use with people with Parkinson's in their life history. And I was able to confirm with 23andMe through their data set that people who had 4 or more courses of antibiotics for strep throat actually had a statistically higher risk of Parkinson's. And I ended up down the path of founding a microbiome company with a microbial ecologist. We collect poop, and we're looking at antibiotic resistance genes and virulence genes and looking at the whole ecosystem. And there are people in the field today looking at ways to outcompete. So if you have a pathogen, They can screen big data sets and look for strains of bacteria that outcompete the pathogen. And so in the way you're talking about looking at it like an ecosystem, and instead of trying to drop a small molecule at the end that's probably not going to solve the problem because it's pushing too hard, is looking at how can you put something back into the ecosystem that can outcompete the weed, as well as look at the terrain that you're building through the food supply.
What you said about Parkinson's has been fascinating to me because as we understand Parkinson's disease more clearly, that it probably begins in the GI tract. And so it's biologically plausible that perturbations in the immunology and ecology of the gastrointestinal tract could be relevant there. So it's really fascinating. And it reminds me to say in part, answer to your question, probably most people know this now, but when people have had broad-spectrum antibiotic exposure and Clostridium difficile steps in to fill the void, it's a dreadful infection in the lower intestinal tract, but the best treatment for it is probably poop because it brings back the normal flora, and so that we are now using that as a therapeutic intervention to try to restore a healthier biome in people. So these are not completely wacko ideas. They really have scientific merit.
Okay.
This has been so interesting that we're going on a little too long. So I'm going to have to start the next talk. Thank you so much, Julia.